Alrighty, I think I have found a way to write on the board that actually records on the video recordings, which is exciting because that wasn't a thing, or hasn't been a thing since I got the new fancy projectors. So I'm gonna be playing around with that a little bit. So, oh, thank you. No, you're fine. Um, so, um, forgive me while we work through that. Today we are going to start. Oh yeah, sorry, me seeing charts a little weird. Um, Today we are going to start talking about cell walls as well as the space between cells, which isn't something people really talk about, and as well as how cells are like joined together, which will lead us into cell membranes. Wednesday, Thursday, and Friday, we're going to start the pre-lab, like lecture-y stuff. So Monday, we will start our like 3-4 day lab. Uh, so that is the generalized plan. My goal for today is for you to be able to explain the role of the cell wall and the extracellular matrix, as well as support your explanations with evidence from that structure. So we will be drawing a little bit today, just as we get through like more complex structure-y stuff. So you may need an extra color or two if you have those at your desk. That is perfectly fine. If you want some color pencils, they're over there, but you'll be fine with just one if you need it. So before I get started, does anyone have any questions, comments, concerns? Alrighty, so this little bit right here in this bold area is what you need. Our extracellular components, so our parts of the cell that are outside of the cell, as well as the connections between cells, help coordinate cellular activities. There we go. All right, there we go. So the coordination of cellular activities is the entire basis for cell specialization. It's the reason why we can have kidney cells and heart cells and lung cells and all of these different types of cells that create special tissues, that create organs, which create organ systems, which come together to create an organism. If cells weren't able to communicate, we would not understand cell differentiation, cell signaling, and cell specialization, which are some really, really, really big ideas in the world that is AP Bio. So that is why we are focusing on these extracellular components so much. All right? All right, so the cell wall is considered an extracellular structure as it is outside of the plasma membrane. So it is considered an extracellular structure. And usually when you first learn about cell walls, it's just plants. It's just plant cells. But cell walls are also found in protists, fungi, and some unicellular eukaryotes, which is cool. So all plant cells have cell walls, but these are some other organisms that do have cell walls. So if we're looking at this bit right here, this plant cell, our cell wall is going to be this dark green along the outside. And if we were looking at this bacteria down here, this cell wall is with this yellow bit over here. It's just another layer outside this plasma membrane to provide some structure and help our organisms function and communicate. So that is our like baseline cell wall. Now the annotations are a little bit different, but that's all right. Bless you. All right, so there are three main functions of the cell wall. Number one is protection. It's that layer of armor around the cell to make sure what's inside doesn't get damaged. Number two is shape maintenance, specifically by preventing excessive water uptake. and we will talk a lot about water uptake next week in our lab. So we want to help maintain our cell shape by regulating the amount of water. There's also the gravity issue, right? Plants don't have bones to help keep them upright like we do. Plants have these cell walls that have the ability to hold them up against the force of gravity. So these are our three main cell wall functions: shape maintenance by water regulation, generalized protection, and holding plants specifically upright against gravity. So in our protists, unicellular eukaryotes with cell walls, their main focus is these first two, but plants have this third special function. All right. So there are two types of cell walls, which is a little bit different than what you may have learned in the past. There are two types of cell walls. There's a primary cell wall and there's a secondary cell wall. So the primary cell wall grows and changes with our plant or organism. It starts out thin and flexible, but as the organism matures, it strengthens and hardens. The secondary cell wall is a layer between the plasma membrane and the primary cell wall that acts as a second, stronger, more durable, extra layer of protection and strength. Not every organism with a cell wall has a secondary cell wall. Usually there's just a primary. But on occasion, certain types of organisms have this secondary bit. So there's a primary cell wall and a secondary cell wall. And we'll talk functionally and structurally about the differences between the two. Questions so far? So we've got a primary cell wall and a secondary cell wall. I mentioned that secondary one is a lot more strong and a lot more durable, and we'll have some examples here in a sec. So, our primary cell wall. These first two things are already what we have written down. This is just duplicate information. So what you need is this last bit right here. And that is how that cell wall matures over time, how it goes from this thin and flexible material to our strengthened one. And one of the ways this happens is through the secretion of these materials that will harden into the primary cell walls. So there are two ways that we can strengthen our cell walls. One is by just hardening the primary cell wall. The second is by creating a secondary cell wall. So some plants will, or some organisms, I guess, will secrete hardening substances into this cell wall, and some will just have secondary cell walls. So this bit right here, this cell wall structure, this orange bit, this is our plasma membrane. This is our cell's plasma membrane. This, green goopy bit up here, that is just the extracellular space called the middle lamina. We'll talk about that in a second. This is what our cell wall actually looks like. It's a lot of cellulose. Remember, cellulose is that carbohydrate that is what makes up plants. It's structural. We can't digest it. A substance is called pectins. A bunch of other types of cellulose. So it's not necessarily like a solid substance. It's a lot of these like interweaving bits of carbohydrate. Our secondary cell walls are often several layers of a strong and durable substance, and this is how we get substances like wood. When we cut down a tree and turn it into some sort of furniture-based thing, right? Or we burn it. What we are burning is not the cells of that tree. Those cells have long died. What we are actually burning is this secondary cell wall. Ooh, go back. So, and it is built up in layers. So, if we've got 2 plant cells right here, this bit right here is going to be our plasma membrane, separating the inside of the cell or the cytoplasm from the outside of the cell. This darker red is going to be our primary cell wall. And this pinkish red is going to be our secondary cell wall, and it will be built up in layers. Questions about primary, secondary cell walls? Alrighty. Oh, wrong way. Old habits die hard, I guess. Oh, there we go. That should save time. All right. So, between cell walls. So, between the primary cell walls of adjacent cells is something called the middle lamina, which is a very thin layer that is super rich in polysaccharides called pectins that will act to glue the stuff together. How many of you, this is kind of a long shot, have ever made like homemade jams or jellies? Yeah. Have you ever used pectins in those to strengthen those? Yes. What do? They make it thicker. They make it more gelatinous. So instead of adding gelatin to jams and jellies, a lot of times they add pectin. Pectin is this kind of sticky polysaccharide or sugar. Remember, polysaccharides are complex carbohydrates that will actually act to thicken up a jam, make it sweeter, and keep it in its more gelatinous state. So there's actually like, we can use pectins just like plant cells use pectins, only we use it for homemade jams and jellies, and they use it to make sure all of those cells stick together. It is not the only way they have cells stick together, but it is one of the ways. There's a wall there. So, if we've got our plant tissue and we wanted to look at our plant cells, this bit right between those cell walls, that is going to be our middle lamina, that kind of glue that holds all of our plant cells together. Whew, doing okay? All right. Nice. All right. Boom. All right. We are going to now move from plant cells to animal cells, specifically talking about the extracellular matrix, which is the area of stuff between animal cells. So the cells in our bodies, the cells in our animal friends' bodies aren't necessarily all like pressed up directly against each other. There's some space in between. That space in between is called the extracellular matrix. And these are their ingredients. Now you only need the stuff that is underlined. I just have some fun facts in there. So Glycoproteins and other carbohydrate containing molecules make up a lot of that extracellular matrix. So, glycoproteins are proteins with glycogens on them or complex carbohydrates. So, it's just proteins and carbohydrates like together in one molecule. And the most abundant of these glycoproteins is collagen. We talked about collagen, I think, in the past. Collagen is one of those like supplements that people tend to take for like hair, skin, and nails. People have collagen infused face products a lot because it is what gives our skin tension. And that collagen forms really, really strong fibers outside of the cell in order to kind of connect those cells together. This bit right here, this fun fact, isn't necessarily write it down, but I think it's amazing. Collagen makes up 40% of the protein in the human body. 40% of the proteins in the human body. Think about how much proteins are active in our body. How many proteins are active in our body at all times? We have proteins that catalyze the reactions that help make our bodies be able to digest food. We have enzymes that carry out I'm blanking, but there's so many proteins that carry oxygen through our blood, proteins that carry out cellular respiration and digestion and metabolism and making our muscles move. We have every single enzyme, so every single chemical reaction in our body has a specific enzyme that catalyzes it, right? We talked about the specificity of enzymes. All of those enzymes put together with all of the other structural proteins in our bodies only make up 60% of the proteins in our bodies, because the other 40% is collagen, which is absolutely wild to me. What happens with these glycoproteins and this collagen is that the fibers form a network of proteoglycans, or molecules that are a protein in the middle with a bunch of carbohydrate chains coming out of it. These proteoglycans are 95% carbohydrate, so the protein part of the molecule is very, very, very small. And that is the big component of our extracellular matrix, or our area between our cells. Doing okay? All right. So there's also fibronectins. Fibronectin is another big ingredient in that extracellular matrix. And it is how our cells are attached to that extracellular matrix through these glycoproteins. Remember, glycoproteins are 5% protein and 95% carbohydrate. And what this does is it binds to surface cell receptors, or cell surface receptors. So there's receptors on our cell membranes, which we'll talk a lot about when we talk about cell signaling. And these fibronectins will bind to those receptors, which we call integrins. And these integrins are built to span the entire plasma membrane. And I'll show you a picture in a second, because pictures tend to help. So we've got these receptor proteins called integrins that are integrated through our entire plasma membrane. And what they do is they transmit signals between the extracellular matrix and the cytoskeleton. So this is how we are connecting from the outside of the cell to the inside of the cell. This is super important in regulating cell behavior, because if one cell is receiving signals that something is going wrong, right, it has to be able to alert the cells around it so they can prepare for whatever is going wrong. So this fibronectin integrin complex is a big way that happens. I promise these are the wordiest slides on this. we've got collagen, we've got other glycoproteins, we've got these proteoglycans, we've like fibronectin, and we have these integrin receptors on our cell walls. And again, you only need what's underlined. So you've got the vocabulary for it. Let's visualize it. I'll give you a second to finish this up. All right. So this is our extracellular matrix, and I'm going to draw it along with you to show you that it can be drawn a lot more simply than we have already. So we're going to start with the plasma membrane. Boop. That's a phospholipid bilayer. This is our plasma membrane. Love it. We talked last week about the different types of cytoskeletal elements, right? And one of those was microfilaments, and those microfilaments formed that kind of complex around the outside of the cell, right? And that is what we are going to draw down here. We've got our microfilaments all along the inside of our cell. And if it helps, you can label this inside of the cell, outside of the cell. Because we're not drawing an entire cell, we're just drawing like a portion of it. So this down here, these are our microfilaments. All right. I'm gonna, didn't need to do that. All right. We'll learn something new. Okay. So then working our way up, embedded in these plasma membranes, we'll talk about all of the kind of stuff that's embedded in the plasma membranes tomorrow, but this one specifically are our integrin proteins. Our integrin proteins, which are just going to be like little blobs on our cell. That's going to be our integrins. Oops, that's an E. These are our integrin proteins. Our integrin proteins are connected to our fibronectins. So our fibronectins, we're just going to have little wispies out here. These are our fibronectins. And these have little branches on them. So little squiggles or little like V shapes will kind of show that wispiness. And we're not going to draw this like proteoglycan complex. We just need to know that this is going to be our fibronectins or our proteoglycans. All right, I'm running out of colors. Let's go grab one. Will it let me? Haha, will it purple? Okay, and finally we have our collagen fibers. And our collagen fibers are going to be a lot larger through here. This is going to be our collagen. Ooh. And then we would have another plasma membrane up here, which this whole complex is connected to, and that's how our stuff is going to be kind of joined together. This is our extracellular matrix. Not too bad. Does anyone have any questions about the extracellular matrix, about what makes it up, about why these things exist? All right. I don't want to erase this if people are still drawing. Is it cool if I erase? All right. Beautiful. Okay, that is our extracellular matrix. We are going to move into cell junctions. Cell junctions. There is one specific plant cell junction, and there are 3 animal cell junctions. And what these do is allow the cells to adhere, interact, or communicate via sites of direct physical contact. So we've got that extracellular matrix, but there are bits in that extracellular matrix where our cells are going to be able to touch and interact. So the reason we have these junctions helps cells get organized, right? Cells are organized into tissues, which are organized into organs, which are organized into organ systems, which will organize into an organism, right? We've got so if we wanted to go backwards, like human being, if we want to focus on our digestive system, right? Then we focus on our organs. So in our digestive system, that's like stomach, small intestine, large intestine, all that fun stuff. And we could look at the specific cells that are going to make up those organs, like our digestive tract cells. So one of the reasons these cells have the ability to organize like that is because of cell junctions, how cells are going to adhere, interact, and communicate with one another. And we'll have some little doodles to go along with these as well. Also, just as a little shorthand trick, this whole sentence could be summarized by cells are organized into tissues, which are organized into organs, which are organized into organ systems, which give us plants and animals. So you don't always have to write out everything word for word. I find that these little arrows are like my lifesaver when it comes to writing down things super quick. So give me just a second. I'll talk about these specific cell junctions. All right. So there is one type of junction in plant cells, and they are called plasmodesmata. We've got some fun words coming in here. Plasmodesmata is the one type of junction in plant cells, or at least the one that we're going to talk about. So if you don't get the word, it's on the next slide. So plasmodesmata, they are perforation in our cell walls, not our cell walls. We don't have these that connect our cells. And cytosol passes through these plasmodesmata, joining these chemical environments, which will essentially create a continuum of cells. So if we wanted to draw this out, I'll just kind of plop down here really quick. We've got our cell wall. It's as simple as that. This is our plasmodesmata. It allows our cells to know how to communicate with one another. What's going on in the cell next to it. A lot of cell communication, especially if you ever take a developmental biology class, a lot of cell communication is done through concentration gradients and signaling molecules. So if something traumatic is happening to this cell and it's releasing a chemical, that chemical can enter through this cell here, and then that cell can react. Either cut it off or prepare for whatever is going on, therefore protecting the rest of the tissues or the rest of the cells in the tissue. That is our plasmodesmata. And again, that will go through our primary and secondary cell walls. Questions here? All right. We also have animal cell junctions, and there are 3 animal cell junctions, and we're going to go through structure and function of all of these. So there are tight junctions, desmosomes, and the gap junctions. These are our 3 types of junctions in our animal cells, so our cells. So we're going to start with tight junctions. Our tight junctions are just where our cells are pressed tightly against one another and bound by these proteins. So we don't need to know the specific protein, but if we have 2 animal cells, they can be held together by this protein or these proteins, and those are going to help perform our tight junctions. I like that one. So we've got animal cell, animal cell, animal cell. We've got these little proteins. That weird P that I draw means proteins that are going to hold those cells very, very tightly together. This creates a barrier and prevents extracellular fluid, so stuff floating around in the extracellular matrix, from leaking across layers of cells. An example of where this is super important is our skin cells. The cells in our skin are divided up into a lot of different layers, and those layers have different types of cells that are going to be held very, very tightly to one another, because we want to make sure our insides stay in and our outsides stay out, right? So holding those cells really, really tightly together is super duper important. So epithelial or skin cells is a big example of where these show up, but they're actually kind of everywhere. So there is still like extracellular matrix stuff going on. So all the stuff that we drew with the extracellular matrix with the collagen and the fibronectin, all of that is still going on out here. There's just little sections where those cells are just sucked closer together. So structure and function of tight junctions. So this one's relatively easy to kind of name. Function connection, some of them are not as kind to us, but tight junctions hold tight on how those cells or hold tight those cells together. Desmosomes are keratin filaments that anchor or have keratin filaments that anchor desmosomes in our cytoplasm, and they act like rivets, fastening cells together in tight sheets. So we've got another way to fasten cells together. So we're going to go back to our kind of handy dandy drawing here. Cell number 1, cell number 2, cell number 3, We've got our desmosomes here in green. And what's happening is we have our cytoskeletal filaments or keratin filaments on one side. So we are again using the internal cytoskeleton of our cells to kind of anchor those together. So those tight junctions are relying a lot on the structure of the cell membrane, but these desmosomes are going to go one step further and use the cytoskeleton of the cell as an anchor. And we have more filaments between the two, kind of stitching them together. These are going to be our desmosomes. Again, we are fastening cells together in tight sheets. Epithelial cells also have a lot of desmosomes because that is where we are really, really worried about stuff being watertight. Doing okay? We got one more. And that is our gap junctions. Our gap junctions are going to function a lot like the plasmodesmata in plant cells. They are made of proteins, and they are going to surround a pore or a hole in the cell membrane where ions, sugars, amino acids, and small molecules can pass through. They provide a channel from one cell to the other, mainly for communication. So we've got animal cell. Actually, let's keep it the color scheme going. Animal cell, animal cell. And we've got this plasmodesmata connecting the two of them. It's kind of hard to see 3D. This picture does a lot better job of that. As you can see, it's not just one of them. These things are peppered throughout that cell membrane. So we've got a nice cross section here, but we can also see on the inside of the other cell that they show up pretty often. All right. Questions, comments, concerns about any of our extracellular